Good morning, everybody. Myself, Professor Ramchandra, Head of Department of Environmental Microbiology, Babasaheb Bhimrao Ambedkar Central University, Lucknow. The detailed mechanism of conversion of methane in different environment are shown in the below figure. The morphology of different methanogens may be seen in the next figure. So this, as I pointed out, that organic compounds is converted into their monomer, and then reduction in short chain fatty acid and alcohol. If there is the sulfate rich condition, then there will be the sulfate reducing bacteria which can oxidize them to the CO2. If there is a low sulfate concentration or condition, then the methanogens will dominate and acetate, formate or hydrogen and carbon dioxide will be produced, and subsequently there will be the utilization of the CO2 and hydrogen production, and in this way two part will may be there. First step there may be the hydrogenotrophic methanogens in one part, and another if there is a acetic elastic microorganism they utilized by hydrogenotropic microorganism. So there will be in the preference of the reaction depending upon the air situation. So this interaction between sulfate reducing bacteria and methanogens exist in the bottom of the sea at the sulfur rich environment. As I pointed out the morphology to see the different morphology of different methanogens, you can see the rod-shaped clear-cut structure regarding the methanobacterium. This is electron photographs, and this is a methanococcus, very spherical structure, and this figure showing morphological view of methanobacterium and methanococcus. The second group of the archaebacteria is thermoacidophiles. They are very unique in their habitat. They are heterogeneous group defined by their ability to grow at a high temperature and low pH. So they are found in hot spring, ocean, sediments, volcanic eruptions. So you can imagine the scene of the hot spring or volcanic eruption. There is a very less pH and very high temperature, and these bacteria survive there. So these bacteria are facultative chemoautotrophs. However, some thermoacidophiles can grow anorganic compounds in its natural habitats. They probably grow as a respiratory chemoautotrophs also. The most common thermoacidophiles are thermoplasma and thermoproteus and thermosulfolobus. So these are the actually important group of the thermoacidophiles. Thermoacidophiles, that is the thermoplasma, thermoproteus, and sulfolobus. So I will discuss about the sulfolobus because in one lecture it is not possible to detail provide all the information about the archaebacteria. So the if you will see the sulfolobus, they are commonly grow in hot spring, and sulfolobus is an irregular lobate spherical structure. The range of conditions over which sulfolobus will grow is fairly wide. They have a temperature range between 55 to 85 degrees centigrade. So it means they can grow up to 80 degrees centigrade or 85 degrees centigrade above than 55 5 degrees centigrade, and pH also remains 1 to 5. It means that is a highly acidic condition, and they have also GC content 60 to 68%. These GC contents actually help the survival in adverse condition of the microorganism. Further, if you will see the cell wall of the sulfolobus, which is composed of principally lipoprotein and carbohydrate containing both neutral sugar and amino sugar, which form a distinct layer outside the cell membrane. Means this sulfolobus has unique property to survive in a very adverse condition in hot spring and volcanic eruptions environment. Geothermal stream or hot water leaches substantial amount of the iron and of the sulfide, which rapidly oxidize to elemental sulfur by oxygen or ferric iron, either chemically or biologically. Sulfolobus rapidly oxidize S2S, but coupling with this oxidation to growth has not been shown much detail. And the principal growth substrate in hot spring is actually elemental sulfur, and sulfolobus grow on the surface of the droplet or crystal of the sulfur, oxidizing it to sulfuric acid, which is largely responsible for acidity of this habitat. I think you people have seen a hot spring or volcanic eruptions site, which has a very adverse environment. And third group of the archaebacteria is a thermoplasma. Thermoplasma is a facultative anaerobes that uses a small number of carbon and energy, and all strains have absolute requirement for unusual growth factor, which can be satisfied by an oligopeptide from yeast extract has been reported so far in the research. Thermoplasma like cell wall, but its cell membrane contains large amount of lipopolysaccharides and glycoproteins, both of which contain mannose as a principal sugar monomer. If you will see the structure of the thermoplasma, you can see thermoplasma, it is a very unique blue color structure as a rod bacillus, and this is known, this is the thermoplasma. And second, this is showing crystals like golden color. This is a sulfolobus, and this another big rod is showing thermoproteus. So this three group of the this bacteria is very interesting, and they have a very unique morphology. Another group under this category is halophiles. Halophiles is actually grow in highly saline environment like a salt lake or brines, harbors a large population of small and distinctive group of bacteria which are immotile cocci or halococcus. They are popularly they are popularly known as a halococcus, and they have a polar flagellated rod. They are known as halobacterium. So this actually group of bacteria which is known as halophiles, they survive and grow in the highly saline environment. Despite the difference of the cell forms, these organisms share a number of properties, many of which are clearly for adaptation to the high salinity and high light intensity of their natural habitat has been reported. The minimum NaCl concentration or salt concentration that permit for their growth is 2% to 2.5 molar, 2 to 2.5 molar, while the optimum range is 4 to 5 mole. The magnesium requirement is also very high in case of this group of the bacteria. The very common example is Hylococcus and Halobacterium. The characteristic feature of the extreme halophile is their contents of red carotenoid, which are the incorporated, which is incorporated into their cell membrane. The carotenoids of Halobacterium have been shown to protect the cell from photochemical damage by the high light intensity characteristic of the natural environment. These bacteria frequently develop as a colored patches on the salted dried fish and hides as a result of the treatment with salt and contains them. They also give the pond in which salt is recovered by evaporation from the seawater their characteristic brick red color. So, if these bacteria is present, there will be the orange color or brick red color appearance. So, the genome of the both Halobacterium and Halococcus contain two components with different percent of the GC value. The majority of the DNA has GC contents above the 66% to 68%, while the minority component associated principally with large plasmid, at least in Halobacterium, has GC contents is 57 to 60%. There is some evidence that plasmid encode the ability to make gas vacuole, commonly it is known as halophiles, and pigment halo, that is known as bacteriorhodopsin. When competent strain of halobacteria are subjected to oxygen limitation condition, they synthesize a chemically modified cell membrane. In anaerobically grown cell, the membrane is red as a result of its high carotenoid contents. A shift to anaerobiosis stops growth and induces the synthesis of new component, that is a purple membrane. So the appearance of color is also characteristic for this group of the bacteria, orange and purple color. The reproduction process and a thermoacidophile and halophile is very also interesting. Halophiles, like all bacteria and archaea, reproduce asexually by binary fission or multiple fission or fragmentation or budding. So, if you will see the color or morphology, this is the purple color and the orange color of this two group of the bacteria, thermoacidophiles and halophiles. And the next in figure, you can see the Hylococcus and Hylobacterium, how they are appearing if they are actually cultured a large amount and they under the microscope. So Hylococcus, you can see the orange appearance of this bacteria, and Hylobacteria is also looking a unique characters. So these are the very interesting, and they survive in the extreme environment. So archaebacteria in this way in the nature is very important, and this play very unique role for the sustainable development of the environment. Thus their importance, if you will see, the importance of the archaebacteria. The importance of archaebacteria can be understood from the following points. The archaebacteria have compelled the scientists to reconsider the common definition of the species. Species are grouped with gene flow within its member. Archaebacteria exhibit gene flow across its species. Archaebacteria are methanogens. They are capable of producing the methane. They survive in very unique environment and they are responsible for degradation of the lot of the organic compounds in anaerobic condition, and they act on the organic matter and decompose it to release the methane, which can be used as a commercial purpose. And they are helpful to control the greenhouse gases also in case of the very unique association of methanotrophs and methanogens. They maintain the diversity in extreme environment for sustainable development of the ecosystem in the water as well as in the on the earth. They maintain the soil fertility and ecosystem in marshy land area. So, if you this is the in brief about the bacteria and archaebacteria, two important group of the microorganisms for their diversity and their role, their structures, their sustainable development of the in the nature for the maintenance of the environment. So, thank you very much. I will be meeting in the next class with some other group of the microorganism, which is a very important in the environment. So, thank you very much.